Okay, so we're going to talk about is aerobic respiration today, which is the supplements what we have on the Zoom and the slides. So aerobic respiration we want to talk about is, first of all, is glycolysis. Glycolysis. So first of all, what is glycolysis? Glycolysis is lysis. You can easily understand is lysis glucose become pyruvate. Okay, so let's draw the cycle. So we have glucose. Glucose will become glucose 6-phosphate. Then become fructose 6-phosphate. Then become fructose 1,6-biphosphate. Okay, start from here. You will see when they become glucose 6-phosphate, there is ATP. Hydrolyzed become ADP and have the phosphate release. And then from glucose 6-phosphate for fructose 6-phosphate, this is just an isomerization. Switch the position of the phosphate. And when you see here, fructose 6-phosphate become fructose 1,6-biphosphate. Here you also have ATP, which is hydrolyzed become ADP and the phosphate. Okay, that's why the phosphate is attached. Now, start from here, there is a branch. Okay, this is going to be a branch. So what's going to happen will be glyceraldehyde 3-phosphate. Oh, so this will be glyceraldehyde 3-phosphate. Then will become 1,3-phosphoglyceraldehyde. Then will become 3-phosphoglyceraldehyde. And then will become 2-phosphoglyceraldehyde, and then become phosphophenols. And finally, it becomes pyruvate. Okay, you need to know this is a branch, what that means. Which means these two lines, there is 2 lines right here. Okay, which indicates there is 2 molecules. Generates through this. Okay, now glyceraldehyde 3-phosphates, they also, some of the textbook said there is an isomerization. That is called dihydroxyacetate glyceraldehyde. Okay, that is they also call it's like isomerization. So usually we don't mention this one too much. Now here there is something happens. When glyceraldehyde 3-phosphate become 1,3-biphosphoglyceraldehyde, they happen something which is NAD becomes reduced to NADH right here. Then 1,3-biphosphoglyceraldehyde becomes 3-phosphoglyceraldehyde. There is an ADP actually with this phosphates becomes ATP. Then finally, when 2-phosphate glyceraldehyde become phosphophenolpyruvate, this is also an ADP with phosphate becomes ATP. Okay, and this is just to switch the position, which is 3 to 2. So there is a question for you. Number one is how many ATPs generate during glycolysis. Okay, now the first answer, lots of you know about this. So at the beginning of this stage, we used the 2 ATPs. Because of the transfer go to glucose 6-phosphate and the fructose 1,6-biphosphates. So they use 2 ATPs. And right here, they generate 2 ATPs. Look at this. Okay, so, and then you have the 2 lines. Therefore, that is 2 ATPs multiplied by 2. So how many ATP generate? 2 ATPs. But be very careful. This results is we assuming this is a closed cycle, which means in anaerobic status, which means there's no oxygen. Okay? So, but the thing here is when we mentioned about fermentation, fermentation is only ATP comes. Where that comes? That comes from glycolysis. So here the 2 ATPs actually is coupled with further fermentation. And you know that when you do the fermentation, these pyruvic acids, they go to lactic acid. They go to lactic acid. And here there is NADH reduced to oxidized to NAD+. This is what happens. This needs to be recycled. So that one can be used again. Okay? So you must be careful. This is what's called fermentation. And this is called homofermentation. Okay? So that's why this 2 ATP comes from here. Now the second question. How about in the presence of absence of oxygen? In the presence of oxygen. That means aerobic. Now what's going to happen? Then we're going to talk about this guy right here. Okay. So when the NAD reduced to NADH2, how many ATP going to generate? 3 ATPs generate. Okay, here is something you need to know. In the old time, when I was student, this NAD reduced to NADH2, we generate 3 ATP. Right now, you may learn it generates 2.5 ATPs instead of 3. Why? Because we are taking consideration of oxygen phosphate ratio. Okay, so that's why you textbook will write about 2.5, but I'm going to be stick to 3.0 ATPs in this class. Just be consistent. So how many ATP generate? We had 2 ATPs used at the beginning. Same thing, we have 2 ATPs right here. We generate, okay? Then how many ATP generate right here? 3 ATPs and multiple by 2 lines. So how many total? 8 ATPs. Okay, now why I want to separate these? Because very important here you need to know. These 2 are generated from substrate level phosphorylation. These 2 are generated substrate level phosphorylation at the beginning. These 2 are generated through oxidative phosphorylation. What's the difference between that? This is simply is ADP adding phosphate becomes ATP. Here we need electron transport chain to help. So which means in aerobic environments, this involved into an electron transport chain. Okay, so here the glycolysis actually have two stories talking about ATP generation. Okay, so we have this pyruvate that comes out. I mentioned in the absence of oxygen, we go through fermentation. Let's say this is a hormone fermentation, we have lactic acid. But how about in the presence of oxygen? This gonna goes to citric acid cycle. Or we say Krebs cycle. This is K. Okay, so what is Krebs cycle? The pyruvate goes there. We have a pyruvate. Okay, we have a pyruvate that goes there. They will go a transition step, become acyl-CoA. Okay, this acyl-CoA actually right here, oxaloacetate, with here will become citrate. And this citrate will become cis citrate. Then goes here, will become alpha ketoglutarate. And then become succinyl-CoA. Then become succinate. And then become fumarate. And become malate. Fumarate. And then become oxaloacetate. Okay, this is called Krebs cycle. This is aerobic, which means you must use oxygen. Now, how many ATP generates right here? This is what we're going to talk. From pyruvate goes acetyl-CoA. This acetyl-CoA CoA will be released. This is a transition step. Happened. NADH. NAD, reduced to NADH. Okay, when you have a cis citrate go to alpha ketoglutarate, this is also happens. NAD becomes NADH. Right here, alpha ketoglutarate go to succinyl-CoA. This is also happened. NADH reduced to NADH2. Okay, here succinate become fumarate. This happened is FAD reduced to FADH2. Okay, and when we look at the last cycle, we also going to see, we also going to see is from the malate to go to oxalate. They also happen is NADH reduced to NADH2. Okay, so how many ATP generate? We mentioned about each NAD reduced to NADH2 is 3 ATPs. So there are 3 right here. There are 3 ATPs right here. There's also 3 ATPs right here. Now, how many ATP for the FAD? FADH2. This is 2 ATPs. Okay, and the new textbook, which is 1.5 ATPs. Same thing, we take consideration of the ratio of oxygen and phosphate. And right here also generates 3 ATPs. Okay? So how many ATPs? We have 1, 2, 3, 4. Okay, which one are we missing? Succinyl-CoA go to succinate, there is a substrate level phosphorylation, which is GDP, becomes GTP. And here, it's equals the ATP generated. Okay, don't forget, all those 1, 2, 3, 4, 5, all right here, which is actually called oxidative phosphorylation. Right here, this is the substrate level phosphorylation. And don't forget about that. So how many ATP generates through here? Which is 3 ATPs at the transition step. Then 3 ATPs multiplied by 1, 2, 3 times happen during citric cycle, and 2 ATPs generated through FAD reduced to the FADH2, and don't forget 1 substrate level phosphorylation, and don't forget this is 2. Okay, so how many we have? This will be 3 add 9 add 3 multiplied by 2. This is equal to 30 ATPs. Okay, so the completely citric acid cycle, Krebs cycle, generates 30 ATPs, which is encoding the transition step. Encodes transition step. Okay, then how many total of the aerobic respiration? So complete aerobic respiration, how many of total they generate? Which is you have 8 ATP right here through glycolysis, and then 30 ATPs generated through transition step and the Krebs cycle. So the total is 38 ATPs. This is an old rule. Okay, you can do a calculation of the new rule if you are using the new one, which is NAD. Go to the NADH2, which is 2.5 ATPs, and FAD reduced to FADH2 is 1.5 ATPs. Okay, you can go ahead to figure out that one. So I'm just help you right here. So this is a whole process, which is aerobic respiration. Now, when you do the calculation, you should understand the concept, the substrate level phosphorylation, oxidative phosphorylation. What's the difference between that? Okay, now, once you see there is a NAD reduced to NADH2, FAD reduced to FADH2, you have 2 3 ATP, 2 ATP generated. This is need electron transport chain. Therefore, what is the electron transport chain? Do you still remember? And we mentioned very earlier, that is caused by NADH, FAD, FMM, FES. Then we go to cyto or COQ, coenzyme Q. So that is COQ. And then we go cytochroma C, cytochroma A. We go cytochroma A3. And finally we go to the oxygen. And that is the electron transport chain. Now when you study this, you're also going to be understanding electron is always go to the donor to acceptor. Now why NAD is a good donor? Because it's very negative based on 2-half reaction of the voltage value. And why oxygen is always at the bottom of this electron transport chain? Because the voltage value is very positive based on the 2-half reaction. The more positive what we say, it is more pretends to be an electron acceptor. That's why it's always at the bottom. Okay? Now this is a typical electron transport chain for aerobic respiration. But how about anaerobic? Anaerobic, there is a lot of the modified electron transport chain, they're not going to generate so much energy. Why? Because compared to the aerobic respiration, from NAD to oxygen, the anaerobic, the voltage distance is much less, or at least less than aerobic respiration. So anaerobic respiration, they will be generated between 2 to 38 ATPs. It's not going to be, some of them say 36 ATPs. It's not going to be as much as aerobic respiration. Okay? Now, last, before I end the section here, I also want to mention this whole process is we talk about is for bacteria. You may say, Dr. Shen, there's a little bit between what I learned in the biochemistry class. What you learn in your biochemistry class is talk about the human being, animal, which is same to the fungi. Okay, Eastern modes, because those are eukaryotes, and bacteria are prokaryotes. So how many in the eukaryotes? It's 38 ATP minus 2 ATPs. The total is 36 ATPs. Now where is that 2 ATPs go? They must be crossing the mitochondrial barrier. They must be carrying on this barrier. They have to think about this way. They generate first in the mitochondria. Okay, then you have to flee away out. Something like you need to, out of the gate, you need to pay the fee. You need to pay the ticket. That is the energy currency. So that 2 ATP is coming from you crossing the mitochondrial barrier. That's why for fungi, for Eastern modes, it is always 36 ATPs because they use the 2 ATPs when they cross the barrier, like they pay the ticket. Okay? So this is the whole process, which is we talk about, complete aerobic respiration. And when you study it, go look at the slides. And I also have a zone, which is a video which is goes really quick. But these are the details for everything. There's lots of information there. You can do a self-study. Okay? Thank you.